This is Lab 17.
So we're still doing hydrolysis here, right? They add water to break bonds, and this time the large molecule we're testing is protein. To see if they can break it down. It's still done externally because they're not big enough to eat a whole piece of protein and digest it inside, so it's external, which means there's going to be an exoenzyme released. And this time we've got a generic exoenzyme name that your lab book gives you, protease, which is a little bit misleading, right? Because lipase was the exoenzyme for lipids, and it breaks down pretty much any lipid it comes into contact with. This protease breaks down protein, but there's not just one. There's not one protease that breaks down all protein in the world. Okay? It doesn't work that way. Every single protein, each individual specific protein, has its own exoenzyme that breaks it down, and not others. Okay, so protease is a very general, very generic term for some molecule that breaks down some protein. But again, just make sure that you know that it's not the magic enzyme that breaks down all protein. Every specific protein has its own specific exoenzyme that breaks it down. Okay? So, generic term there. We're going to be looking at two specific proteins in this lab, gelatin and casein. Start with gelatin. That's our first protein that we're going to give to bacteria and see if they can break it down. A little background on gelatin. This is the stuff in Jell-O that makes it solidify in the refrigerator. Anybody know where it comes from?
Animal bones.
Animal bones and clothes. Right? They boil it out and extract it. Just like if you've ever made chicken soup at home, right, by boiling a whole bone-in chicken or chicken hearts with bones in them. You are extracting the gelatin from the bone, and then when you put that chicken soup in the fridge, it gets kind of cold and jelly-like as it sets up. So same process. None of that's going to be on your quiz. I just think you should know. Gelatin's not vegetarian. Explains why they give it to you in the hospital, though, right? It's full of protein and easy to chew. You can't chew a steak if you're sick, but you can swallow Jell-O and get plenty of protein from it. Okay? Nothing at all wrong with it. Perfectly good nutritionally for food. But at any rate, that's the gelatin we're going to be giving them. Petri dish again. The agar is gelatin nutrient agar. Remember, I'm a stickler for the names on quizzes. I want spirit blue lipid agar for the lipase test, gelatin nutrient agar, all three words for this test. Basically, we just make some of that powdered gelatin in with the nutrient agar when we made it. And that's your first Petri dish here, well, my right, your left, I guess. The gelatin nutrient agar. You're going to do the same thing that you did last time, divide it up into 3 sections, right, with your pen, marking it on the back on the agar side. Got a control section. You've got to read your lab book to see which 2 bacteria you need. If you look at your materials page, there are going to be 3 bacteria listed. You're not using all 3. You've actually got to read your procedure for this one, right, to see which 2 bacteria you're putting in the gelatin plate label, right, which 2 bacteria you're putting in there so that you know later. You're reading that from your lab book.
Do we need to know each one of the bacteria for each one of the tests?
No, I'm never going to ask you which bacteria did you use for the starch test or the gelatin test, or no. But once you find out what they are, aseptic technique, right, flame your lube, the opening of the tube, and you're making one short straight line, just like you did last Wednesday, of that bacteria in its specific section. And again, that's all you do today, right? Inoculate it, get it in the incubator, same time and temperature, right? 37 degrees, 18 to 24 hours. And then after incubation, because gelatin is sort of a tan color on its own, we're not going to be able to tell if they've eaten it or not. We're going to have to add a reagent. And the reagent that we use is different than the one in your book. Okay? Trichloroacetic acid. This is the actual reagent that we use. Your book says to use acidified mercuric chloride. If you learn acidified mercuric chloride and you were to write that down on a test, I wouldn't mark you wrong, because that's what your book says. But we're trying to be a mercury-free institution and not use anything with mercury in it. So this is our substitute, trichloroacetic acid. And you can certainly learn that one for the quiz too and get the right answer. trichloroacetic acid and acidified mercuric chloride, they do the same thing. What this reagent will do is precipitate proteins. Okay, precipitate, makes them come out of solution so that they're not dissolved anymore. Precipitate means that they fall out of solution or they clump up out of solution. And they turn sort of a cloudy white color when that happens. Okay, so precipitates, they fall out of solution, they clump up, and they turn a cloudy white color. So when we come back next time and we add trichloroacetic acid, what's going to happen in the control section of the plate?
Nothing.
It's going to clump and turn cloudy and white, right? Because it's got gelatin protein in it. There's no bacteria in there to break it down. So when we add the reagent, all the protein in that section should clump up and turn cloudy and white. All right, so if this was the positive bacteria, and it's releasing the exoenzyme, what's going to happen to all of the gelatin around it? It's going to be broken down by the exoenzyme. So when we add the trichloroacetic acid, is it going to clump?
No.
No. We're going to have an area around the bacterial growth that has no white color. That's your positive result. No white color around the bacterial growth.
What did you say the exoenzyme was?
I didn't give you the name yet.
Oh,
That's a good question. Let's do that. Exoenzyme is gelatinase. Is it specific for gelatin? Right? Gelatinase is the exoenzyme that breaks down gelatin. It doesn't break down any other proteins. So positive result again, no white color around the bacterial growth. And that means the bacteria produce gelatinase. If this is our negative bacteria, it's not producing gelatinase. We add trichloroacetic acid, what's going to happen?
Cloudy and white.
It's going to turn cloudy and white. Yep. So negative result. There will be a white color around the bacterial growth. Now I will caution you with this, and I'll remind you again when we actually look at them on Wednesday. The bacteria themselves have protein all in their cell membrane and cell wall. They're covered in protein. So when we add trichloroacetic acid, that little strip of bacteria that you can see is going to turn white because the proteins on their surface are going to clump up. So it's not the color of the bacteria themselves. They're all going to turn white. It's the agar around them that we're interested in. Okay, so pay attention to that. That's why I said around the growth, not the bacteria themselves. All the bacteria are going to turn white.
Is that almost like another halo in a way?
Yeah.
Okay.
Yep.
But a
But
white
again,
one.
just be careful learning it as halo because I need to know that you know what color it would have been. The halo for starch would have been black. The halo for this would have been white. So if you just say halo, I don't know that you know what it would have been if it had been negative. Okay. So yeah, it's important to note the color. Okay, this one's in your photographic atlas, page 73, the gelatin test. So if you want to look it up later, just write it down, page 73 as a gelatin test. Still in Lab 17, we're going to look at a second protein now. So gelatin was one. Second protein that we're going to look at is casein. And this is the protein in milk. Casein is the protein in flesh.
Excuse me, thank you.
Protein in milk. It's what gives milk the white color that milk has. It's because of the casein protein. So it already is white. So you can actually kind of see the difference. If I hold up the gelatin plate and the casein plate, the whiter one has the milk in it. So it's the casein that makes it white. So that's the protein we're going to give them. And what we're testing for is to see if they make the exoenzyme for breaking it down, which is called casease. So that's the exoenzyme that destroys casein. But again, not every bacteria can produce it, right? That's what we're trying to find out. Do they make casease or do they not? So that agar that I just held up that has casein in it is called skim milk nutrient agar.
It's funny,
Yeah,
skim
you
milk.
think it's funny, you should try and make it. That powdered milk that you can buy in the grocery store,
Yeah.
you mix it with agar and heat it up. It almost always gets coagulated in the autoclave at high heat. It's not a fun one to make. It smells, but it's already made for you. You don't don't have to do that. Skim milk nutrient auger, again, I'm a stickler for the name, make sure you get all 4 words there.
Skim milk.
So there's already casein in, right, with the auger.
Right.
Same thing, you're dividing the plate up into 3 sections, reading your procedure to see which 2 bacteria you're supposed to use for the casein test. And you have a control section of the plate again. And again, it takes me longer to tell you than it will for you to do it. You're just adding aseptically 1 short, straight line of bacteria in each section. And we're not trying to get isolated colonies here. And you incubate, as always, same time and temperature. There's no reagent for this one because casein already has a white color to it. We're not going to have to add anything for color change. If this were the bacteria that produces casease, while it's in the incubator, casease gets excreted, all of the casein around it gets broken down, and so what happens to the white casein color?
It's eaten, so
Right,
it's
it's going to
clear.
be clear, right? No white color around the bacterial growth. Positive result here.
No white color.
No white color around the bacterial growth.
Positive.
Yeah,
So that means the casease was produced.
absolutely. Okay. So what's negative going to look like?
Regular white.
Has white color, right, around the bacterial growth. And that means it did not produce
Did not
casease.
produce KCH.
Good. And that one's on page 62 of your photographic atlas, the casein test, page 62.
Okay.
Alright, so that's all lab 17. We're doing one more lab today because these are so short.
Is there-
Lab